Okay, I'm really sorry that Cindy can't be here. These are her slides, and the team that put this together was a phenomenal team, and Cindy has a lot of passion for this, so this is her show. She had no disclosures, and I think you just heard mine. So the workshop background was that many studies of the intestinal microbiome, whether in vitro, in animal models, or in humans, report only minimal information on dietary components, even though there is substantial evidence, as you heard previously, that diet modulates the composition of the microbiota. Although many aspects of the microbiome study show improved quality control over time, this has not always extended to diet. There's lots of aspects of measuring the microbiome and relationships to health. It's a very, very tricky area, but this workshop really was to focus on diet. So the workshop objectives were to improve the rigor and reproducibility in research on the colonic microbiome, to identify important dietary information that should be reported, to identify the many parameters to consider in the design of studies, particularly for clinical studies on diet and the intestinal microbiome, and very importantly, to publish a summary for the research community in a peer-reviewed journal that can really help increase the strength and quality of the papers to be published in this area. So there were 4 sessions, and the first session was focused on the characterization of dietary fibers and other nutrients that feed the microbiome. And the other 3 sessions focused on animal models, in vitro models, and clinical studies. But the fact that fiber is so important as the key material that the microbiome utilizes, it was very important to focus on fiber. And fiber is perhaps the most chemically complex food constituents important in human nutrition. The speakers did an excellent job of outlining how complex fiber is. And it explains the issues inherent to its definition and analyses. We have lots of different definitions of dietary fiber. We need better information on how foods and food additives lead to shifts in microbial digestion of glycans, I'm sorry. So the next session was animal models, and you could see the speakers, an excellent team of speakers for all the sessions. So the key points here was, although the AIN 93 diet, which is the chow diet, is considered the gold standard in nutritional studies that use rodent models, it contains no fermentable dietary fiber, which is just an amazing factor when you look at how important fiber is with the microbiome studies. So that can be advantageous for what you want to add in, or disadvantageous for representative of a real diet. Microbiota in mice shows circadian rhythm, which is shifted by diet. So what we need to know is this the same in humans? And many dietary constituents can modulate the composition and activity of colonic microbiome. So these include polyphenols, proteins, omega-3 fatty acids, and emulsifiers and other grass substances may affect microbiota and inflammation. We're in the early stages here. It's animal models, and I think here another very important consideration is, are we using physiologically meaningful doses? Non-rodent models were also discussed. Rodent models are the most prevalent, but non-rodent models are possibly better models, but not validated. So with animal models, some of the unanswered questions and research gaps are notobiotic animals with defined microbiota valid models of animals with natural microbiota? And what about wild type animals? And Gary had spoken to some of his work in that area. What is the optimal diet for animal studies? As is in the case for most things, I don't think there's going to be one optimal diet. What are the causes of variation in both animal and human microbiota? And I think that we heard at the conference that 60% of the variation in animal microbiota can be explained by diet, whereas in the human microbiota, it's 10%. Again, the complexities. So gaps. Some of the gaps are we need better tools to study how individual strains colonize the gut, and we need adequate data on other organisms, as was mentioned earlier, such as archaea, fungi, viruses, parasites, and bacteriophage, and many of their functional capacities. So this is very important, and this is an area that goes beyond diet. So session 3 focused on in vitro models, and this was really an interesting session in learning all of the considerations. I mean, in vitro models are excellent for generating hypotheses, and there's many different types of bioreactors, and I think the multi-stage ones have the biggest benefits, but you have to look at how you can remove metabolites that go as the studies progress. There's so many complexities. They can be good for single organisms and complex ecosystems. Defined communities allow precise understanding of ecosystem functions, and in vitro systems certainly make it easier to apply treatments, obtain samples, and are accessible for omics sampling. Gaps are source of the inoculum, feces. I mean, given the challenges with diet, person to person and intra-individual variation, huge challenge there. Knowledge of the diet of the fecal donor and nature of the nutrient supply. The final session focused on human studies, a great group. Key points, large differences that we heard in hunter-gatherer or agrarian versus Western diets in microbiota and metabolome. Controlled feeding studies are useful for investigating the effect on gut microbiota and host response, but they have limitations such as access to tissues of interest, mostly short-term outcomes, and challenges establishing causality. Studies with fiber mixtures suggest interventions can support GI microbiota during antibiotic treatment, very important. And fiber recommendations should be based on colonic microbiotic requirements, not cardiovascular health benefits. So they'll change in paradigm. So human studies, unanswered questions and gaps. Does diet affect recovery of gut microbiota after disruption with antibiotics and infection? There's some good research starting here. Why does diet explain about 60% of the variation in the gut microbiome in mice, but only 10% in humans? So gaps, better methods to determine dietary exposure. This came up as one of the key emerging issues for consideration. There is no biomarker that reflects dietary fiber intake or status. Many issues are ignored in studies of the human intestinal microbiota, such as feces collective baseline microbiota, lack of basic information on how transit time affects composition of the colonic microbiota and prebiotics. No prospective cohort studies linking change in the fecal microbiota with health endpoints. So in the conclusions that in the absence of clearly superior dietary approaches for specific research questions, the main recommendation is to provide as much detail as possible about feeding and housing conditions of test subjects so that other researchers can reproduce the work. Characterization of a core healthy microbiota, and more usefully, their metabolic functions remain elusive. And the emerging field needs to move from association to causality, which will be catalyzed by knowing it as much detail as possible about what is ingested, how it is metabolized, and the health consequences derived from these processes. And that's Cindy's thing. And I would say one thing we saw from everybody else's talks was the huge group, I think, that works on all of these studies. And I just want to make a plug. It's really, really important to involve food scientists and nutritionists in the teams to help design the dietary components. But this workshop was great. It was, led by NIH with USDA and ILSI North America support. So thank you.